Welcome, good afternoon, everyone. The topic of this session is our community ready for gene therapy? My name is Shruti Mitkus. I will be moderating. I am the Director of Patient Services at Global Genes, and I'm also a geneticist. And I am delighted that we are having this discussion. Just to date myself a little bit, I've been in the field of genetics for many, many years, and even a decade ago, we would not be having this conversation about, is our community ready for gene therapy, particularly in the rare disease space. So I am very excited that we get to talk about this today. I'm going to be giving our wonderful panelists a chance to introduce themselves now before we dive into the content. Starting with you, Ashley.
Yep, this is on. Great. My name is Ashley Point. I am first and foremost a mom to a rare disease boy. His name is Davis. He is 10 years old. He was diagnosed by a geneticist at about 16 months with KDVS after missed milestones, respiratory infections, and a seizure. And so we are excited today to be a part of this possibility of gene therapy, right? I mean, that's every patient advocate's dream, is to find a treatment or a cure for your kids. So I'm the president of the Koolen De-Vries Syndrome Foundation, and we're just happy to be here.
So my information's actually wrong on there, so I'll just verbally update it. So I'm Matt Fuller. I'm Vice President and Head of Gene Therapy Research at Ultragenyx. I've been with the company for about 7.5 years, focusing exclusively on program research for gene therapy and then also platform research so we can try to make this drug as cheap as possible so it's actually accessible for all the patients.
And my name is Ray Belanger. I'm a genetic counselor at Boston Children's Hospital, specifically within the Rosamund Zander Translational Neuroscience Center, and very happy to be here to talk about readiness for gene therapy.
Great, thank you. Before we dive into the content of this session, I'd like to set the stage a little bit. As we know, about 75 to 80% of rare diseases are genetic in nature, and quite frankly, I suspect the rest of the 20% probably have a strong genetic component that we just don't know about currently. And so it's perfectly reasonable for patients, caregivers, and organizational leaders like yourselves to ask the question, how can I make my disease ready for gene therapy? How can I have my disease be an attractive candidate for development for the gene therapy pipeline? And those are perfectly reasonable questions. But it's easier said than done. There are a lot of steps to be taken to lay the groundwork to be ready for gene therapy. And so a couple of years ago, Maya Chopra's group at Boston Children's developed a comprehensive framework to evaluate the research of a particular disease and a stepwise process to things to consider in research and development to prioritize a disease for gene therapy. And so after meeting our panelists and talking about it, I think the way we've structured this particular session is we're going to walk through this framework. It's called Gene Target, with the idea that how can you, as community leaders, prioritize your research plans, because the reality is funding is not unlimited. You have to be able to prioritize certain aspects of preclinical work on your disease before we can get to the step of gene therapy. So we're going to walk through this framework called Gene Target. Ray's going to walk through it, and we've structured it so that we're going to pause periodically after every few slides, and we're going to get perspective from both an advocacy organization perspective with Ashley, as well as industry's perspective, because they might not be exactly the same, and we'd like you to leave with both perspectives at this point. So, Ray, with that, I'd leave it to you.
Thanks, Shruti. So as Shruti describes, the folks at the Rosamund Zander Translational Neuroscience Center, led by Dr. Maya Chopra, put together a very comprehensive framework to think translationally about a given gene or disorder. So we're going to walk through a specific example looking at Koolen De-Vries syndrome. Just very happy that Ashley's here. And this is very much a work in progress. You know, we're currently working on some revisions of the framework. We're always trying to make improvements. So to get feedback from a PAG representative as well as industry where we see that, this framework could have implications. I'm very excited to hear both of your feedback, so very much appreciative of that. So the first paper on gene target was published in 2022. And as Shruti alluded, it's a way to think translationally about a gene disorder and specifically look at clinical trial readiness. Kind of within that, looking more deeply into therapeutic development for gene therapies. So while it's geared more towards rare monogenic neurodevelopmental disorders, which is what our research center focuses on, we think there could be applications wider than that. So I'll jump into some exact details here. So gene target is actually an acronym that stands for all of the things that you can see in the table here. And it's broken down into 4 major domains. And within those domains, we've weighted them, we've allowed the framework to work to be able to generate a score. And each of the 4 domains add up to 10 points, so we weight them equally in the measure. And the total score that you can get is 40. So looking into the 4 domains, we like to look at clinical considerations. So these are things like how early a diagnosis is made for a condition. Is the natural history of the condition understood? Are there endpoints that are validated that can then be thought about in a clinical trial sense? And if there's availability of other safe and effective treatments for that condition. Looking into some more genetic components, we want to understand the genetic mechanism of the condition, and then also looking into specific dosage changes and dosage considerations. For preclinical considerations, we want to look at different tissue expression and gene expression within those target tissues and whether or not we have the biological tools to be able to target those tissues also at the right time during development. And then we also want to look at reversibility information, if tests in model organisms, if there's been any demonstration of reversibility of certain phenotypic features. And then lastly, we also want to think about the ethical considerations that go into translational research, if these principles have been considered at all. And then also seeing if the target population that you want to work with is accessible and engaged. So all of these things together kind of make up what we think are the key components of the translational pipeline. And this slide is a view of kind of where we're currently at with research surrounding gene target. Myself and my colleague Abigail and a couple of our colleagues at the center have been working through evaluating a number of genes to make refinements to the framework and potentially identify limitations. And we're making revisions to see if we can make this framework a bit more comprehensive, capture more of the many, many nuances that go into evaluating a translational pipeline for a gene and disorder. And then also taking in some additional points beyond gene therapy approaches, things like medicinal chemistry and small molecules. And then we also want to see if this framework performs well if you have people of different backgrounds doing a gene target evaluation. So we're looking at some inter-user variability assessments, having a number of raters score the same set of genes to see how similar those scores are. So with that, I'll kind of dive into the example here with KDVS. So the first component is understanding the genetic mechanism of the disorder. And you'll see here each score will have essentially a Likert scale, or kind of a Likert scale, where we have the numerical things on the right, and then each score has a specific description associated with it, depending on the level of evidence that we can find to support these scores. So for KDVS specifically, we know that roughly 60% of individuals who have the disorder, this is caused by a microdeletion at 17q21.31, while the remaining 40% have variants in a gene called CANZEL1. So when thinking about a gene therapy, if you have a disorder that's caused by a copy number variant such as microdeletions and microduplications, this could make a gene therapy approach a bit more complex because you're focusing on a genomic region that can contain multiple genes versus a singular gene that you can target. So based on that information, we've scored this as a 2, given that there are multi-gene copy number variants associated with the cause of the disorder. Next, early diagnosis. This was actually one of the ones that we've made revisions on, where we know in rare disease, coming to a diagnosis, it can be a very long journey. So we didn't want to necessarily score a lower number because of that. So instead of just focusing on when that diagnosis is made, we also want to consider when the clinical presentation is and how early those features of the condition are presenting. So in the case of KDVS, we know that these features typically present in childhood, early childhood. So instead of focusing just on the diagnosis, we want to look at the clinical presentation as well.
I want to say at this point, it's really important to highlight early diagnosis. I know we hear this a lot in the rare disease space, but often it can take many, many years to get that diagnosis. And so once again, highlighting early diagnosis at the genetic level, be it through rapid genome in the NICU setting, or other ways that even advocacy organizations can enable their communities to get to a diagnosis quickly, is really the starting point to be able to consider gene therapy when we're going through a paradigm like gene target. So I just want to highlight that early diagnosis is really key.
Moving on to natural history. The gold standard for natural history data is longitudinal information, so we can see how patients evolve over time. And looking specifically at KDVS, we know that there's cross-sectional studies that have been published looking at large cohorts of patients to describe their features. When thinking about longitudinal information, it's difficult to know exactly how that information is going to be published. So an important note about the gene target framework is that it's working off of information that's publicly accessible, so things that you can find in databases like PubMed, other places where you can review peer-reviewed articles, but reaching out to your connections. If you have a connection with a patient advocacy group that you're doing a gene target for their condition, they might have some additional knowledge that would influence your score here. So I wanted to pause here and see from the advocacy perspective how you're publishing that information and kind of what public-facing information you want to have available to researchers who are looking into potentially developing therapeutics for your condition.
Yeah, I think, as you kind of previously stated that sometimes the data is not published, right? Like natural history study is going on in longitudinal study, and it's not really been published yet. So we have some studies that were done in the Netherlands that have been published and some information on that, but also working in the US to get that set up as well.
And also, Matt, from an industry point of view, how important is having that longitudinal data or natural history data important for a industry to consider?
So the more you have, the better, right? That's always going to be good. But you mentioned when we first sat down that there's a balance between how much you can spend and what you can accomplish. And so if you were to go to a company and have no research, know nothing about the gene, but you had this natural history study, I don't think that's a good balance. I think you really want to try to balance both of them. Keep in mind that there's more than one way to do a natural history study. You can do a retrospective one, you can do one that's ongoing, you can partner maybe with KOLs or a hospital. You just need to make sure, I think as you had mentioned, Ashley, that the data is publicly available. That's the most important thing, because there have been a lot of natural history studies conducted that are essentially behind a paywall, and you may or may not know it. And so we're wasting a lot of people's time in their lives doing the things again.
I think that's a very important point. And also I want to highlight, it's really important that the patients own the data. It's publicly available, but the patients get to decide who gets access to that data. Yeah.
Kind of leading off of natural history as they inform endpoints, we've seen kind of anecdotally across, specifically neurodevelopmental disorders, that endpoints are a challenge for a lot of folks. In the rare disease space, it's a common hurdle for groups. They're difficult to define because you're looking, specifically at cognitive measures, you're looking at behavior, and quantifying and looking at these in a structured way is really difficult. In addition, endpoints are best informed by longitudinal natural history data, and those protocols are difficult to establish. So kind of as we've looked across many neurodevelopmental conditions, this has kind of been a common theme we've seen so far.
It's always a challenge, right? I mean, not just neurological, pediatric onset neurological conditions, but I think across the board, picking the right endpoints is always a bit of a challenge. And so I'm wondering, are there any surrogate, or biomarkers that we can use, not in the absence of endpoints, but that could also help us determine that? And Matt, would you have any comments on that one?
So this might be a non-answer, but I think what's most important is for the patient groups and the patients to tell whoever they're working with what is a valuable endpoint to them. Because if it matters to the patients, then it's going to be much easier for whoever's running that trial to explain to the FDA why it matters. Now, you might have to get creative in how you measure, quantify, that sort of stuff, but if it truly is critical to the nature of that disease and what needs to be addressed, then I think that's what really makes it powerful. It just might take some creativity to get it to, be acceptable.
And would you say, I'm just going to ask this question, would you say having more endpoints and more varied endpoints is better or worse than having more defined endpoints, or?
So I will say I'm not a physician, so it's a little out of what I know. I don't think you want to, throw the kitchen sink at it. I think if you just have one or two, that can be complicated unless they're really well established and, maybe it's a disease that people are really, really familiar with. Otherwise, I think what you want to try to do is get a mix of things that are a little bit more, let's say, accepted or more easily accepted by a health agency. And then if there are things that you think are really, really important, push for them. And they can hopefully sort of balance each other in that assessment.
So this next section gets at more of the preclinical considerations when thinking about clinical trial readiness. So for this score, we're looking at target tissues, so trying to identify the cell type. In the case of neurodevelopmental disorders, oftentimes it's neurons. We want to know specifically what neurons are affected, what regions of the brain are affected, what those cell types are. And then also seeing if there's any biological tool that is effective at targeting these tissues. And then there's the timing aspect. When in development is gene expression occurring? When do we need to intervene to have the most therapeutic impact? So for these preclinical studies, they can be difficult to prioritize. So I'd be interested to hear, from the KDVS Foundation, how this prioritization happens, and if you have any advice to younger PAGs who, are just getting started developing a research plan.
I appreciate you showing our strategic research plan. That was one thing we definitely prioritized when, about a few years ago, we said, okay, where are our gaps? And we had an outside group come in and really go through all the data, all the published reports, talking to the families, what are we missing? And that really showed us our gaps, and so then we can go back and say, okay, this is what we're missing. We need to really focus on these aspects moving forward. And then also talking about younger PAGs, and what I would recommend would be to collect and store that data, whether it's seizure reports, whether it's different aspects of your syndrome, but store it in a way that is protected, whether it's HIPAA compliant, it's GDPR compliant, it's in REDCap, get your IRB, do that stuff that you do early, again, so you own the data and you have that data, but it's also protected because you would hate for something to happen with that data.
I think it's also really important for quote-unquote smaller PAGs, patient advocacy organizations, to to learn from larger organizations that have gone down this road, that perhaps have like a robust scientific advisory board that's able to tell them, you know, with the limited budget and on research funding that you have, prioritize this particular preclinical study or what have you. So I think we cannot underestimate learning from others and building capacity by learning from others that have gone down that road. And it doesn't have to be in the same disease, even disease group as what you're dealing with. I think there's a lot of knowledge that can be shared across when it comes to a research readiness plan. Yeah.
Yeah, and I'm happy actually you brought up that this, you know, came from your strategic research plan, and this just kind of highlights more making this information publicly accessible to people who are potentially interested in your condition. There was some great information in there, and I pulled a direct quote from that, which helped, you know, inform the score that we generated for KDVS. So having that information available either on your website or posted on your social media platforms is really beneficial. The availability of other treatments is pretty straightforward. And the scoring here is a little bit counterintuitive because we want to prioritize conditions for which there aren't you know, disease-modifying treatments available. So for the case of KDVS, given we don't have any approved disease-modifying treatments at the moment, that would generate the highest possible score in this section. For reversibility, this is kind of almost a proof of concept for some of the therapeutic approaches that may be in the works. Looking in model systems such as iPSCs or animal models where you're testing, you know, maybe potential compounds, molecules, other treatments, to see if you're able to address some of these phenotypic features that are associated with the condition. So for the case of CANZL1, you know, we found this paper where 13-cis-retinoic acid can reverse some of the features that we see kind of on a biological level and then on a larger level in a CANZL1 knockout mice model. In addition to establishing reversibility, you need to understand, or it's important to understand the temporal window as well. And it seems like based on, you know, the information that we were able to find, that temporal window piece still needs to be fleshed out a little bit more.
And I think the reversibility is a great thing, but I would like to hear from you, Matt. You know, what threshold of reversibility would be attractive to the industry? I mean, how reversible should the phenotype be?
Yeah, there's two things that I think of there. One of them is, again, what's important to the patient. Because maybe what's really important to them can't be reversed or can only be reversed to a point, so we just need to be really realistic about that. But then the other is, I might have forgotten what the other one is. Yeah, I don't know, I can't remember. I guess it's sort of, is it a really basic function or is it more in a model system? And so I guess what I mean by that, because that wasn't very clear, is sometimes it's important to say, okay, it's a kinase or whatever. We can reverse that kinase function, and you can show that in cells. But that's not enough to really demonstrate true reversibility. So you also need to be able to have a mouse model or some large animal model and say, okay, let's say neuron, you know, interactions or whatever. We can show that when we treat with this, we actually reestablish something that's more on a global scale. So the more data you can show, the better, which I know is obvious, but I would start with cells of some sort, especially if you can use iPSC-derived cells and have something that is hopefully, you know, mutated or knocked out, and then you're adding back into that, but then go into a model. And then hopefully those actually do go back to what matters to the patients.
And it sounds also like it's important that it's quantifiable, and it doesn't have to be magnitudes of you know, reversibility, seeing it on a scale of magnitude, but certainly quantifiable, and if it shows an effect in cell lines or...
Yeah, and in a cell line, right, it's going to be more quantifiable. Let's just pretend you're in a mouse model and it means they go through a maze 5 times faster. That's meaningful. It also might just be that, you know, they stop having seizures every so often. So I think you have to be it's really going to be dependent upon the disease that you're working on, because it needs to reflect what's truly happening to those patients and where an improvement can be made for them.
Moving on to being tolerant to dosage changes. So dosage refers to the number of copies of a given gene that's present in the genome. So typically we have 2 copies of every gene. And we know that there are some conditions that are caused by one of those copies not working properly. These are known as haploinsufficient conditions. There's also disorders that are caused by having too many copies, 3, for example. And we want to define what that therapeutic window is when considering what approach you might take at addressing this dosage. So in the case of KDVS, we know that this is a haploinsufficiency condition, meaning that if one of those copies is not working, then you have features of the condition. But looking deeper into the literature, we see that this genomic region, if you duplicate it instead of delete it, we also see features of disease, not necessarily the same thing as KDVS, but they present in a slightly different way. So when thinking about therapeutic approaches of, for example, in a haploinsufficiency condition, if you want to upregulate the working copy of the gene to kind of compensate for the one that's not working, you need to be mindful that if you turn up the volume a little bit too much, this could also lead to health problems.
Which it's kind of interesting here that it's that when you do the microduplication, because that they have behavioral problems and poor social interaction, because KDVS kids are known to be very friendly, outgoing kids where they'll go to anybody. And so it's interesting that it, when you add more of it, they struggle with social interaction.
Right, I think it's like the Goldilocks effect, right? It has to be, and that's for a lot of genes, right? Not too much, not too little. Just right. And again, it goes back to really understanding your disease at the genetic level, which is what we started with, to really know how much of that dosage is tolerant in your disease. Yep. And
you actually reminded me what I forgot. So for the reversibility, the therapeutic window that you have highlighted there is really important. There are some diseases, I think CDKL5 deficiency disorder, where it's thought that reversibility is probably a pretty long window. There are some diseases where we know if you hit this age, you can't reverse anymore. And so if you can define that, that's really important for convincing whoever you're working with to develop a therapy, because it lets them know how many patients are actually treatable and how many people they can help. And the other reason is that way you can understand the model you're using, because maybe you're using a mouse model that's, 9 months old, and that's so far outside of the window that may or may not be a good outcome. So again, the more you understand about your disease, the better.
Coupled with what patients feel is important. Yeah. So these last two considerations are focused more on ethics. So when we're thinking about a favorable risk-benefit ratio, we think about things like how severe the condition is, if there's any available treatment, how early the onset is. And then we also want to consider how generalizable a therapeutic may be, at least from the perspective of the research center that I'm with at Boston Children's. We like to focus more on therapeutics that could affect as much as the disease population as possible. Obviously, there's some nuances to that. But in the case of KDVS, given that we know that there are roughly 60% that are due to the microdeletion and a genomic region instead of just a single gene, this may limit some of the generalizability across that population, given the different biology of that disease presentation.
So for the KVDS community, Ashley, could you talk a little bit about, and this is going to be different for every disease community, of course, but from your perspective, what is the risk-benefit tolerance in this community, and how do you weigh that when you're looking at something like gene target?
I think that's an interesting question because it is so different for everybody. Like for my son, we feel like we're pretty well controlled, right? Like our seizures are under control and he's talking and he is able to walk. But then there are other kids in our community, 5, 6, 7 years old, not potty trained, nonverbal, still having uncontrolled seizures. So I think it's going to vary, as I'm sure with many people who have, a spectrum of disorders. It really will. It'll be interesting to see which families jump on it, and others are kind of like, I don't want to rock the boat. It's too much. So, I'll be interested.
That's a great point. And same question for you, Matt. Does that look different from an industry perspective?
So, not to be too qualified, I think it depends. If you work with a company that's incredibly conservative, they might only want to do things that are just like home runs. And so you just need to know that going in. The way that I hope companies would approach it is, what is the risk benefit that the patients and that their caregivers are willing to take? And so, I think you described that, but I work with Courtney sitting over there on the Koolen Syndrome Research Foundation, and their kids live long, happy lives, but they lose their ability to see. So they don't have the same risk tolerance as, say, someone that has spinal muscular atrophy, where they know something devastating is going to happen. So those groups are going to take very different risks, and my hope is that if they were working with a company, that company would weigh the risk-benefit in a very similar way, because it really demonstrates, I think, where you need to put your resources and how long you have to do that.
And I think it's really interesting also to talk about how can it be generalizable for the population? Is it just a N of one therapy, or is it applicable to the entire community? And I really appreciate that that's built into this gene target framework, because that's something to really consider when you're thinking about gene therapy for a particular condition. How generalizable can it be for that entire disease community? Yeah.
And lastly, we want to see how engaged and accessible a population is. The Koolen De-Vries Foundation is a great example of a very engaged patient advocacy group. They have a fantastic website. They have a contact registry on their website. And how accessible your patient population is, I'm sure you've heard this throughout the conference, is really important, especially when considering clinical trial readiness. You can do all the work to get to a clinical trial, but when you get there, if you don't have patients who want to enroll, if you can't galvanize your community to want to be a part of these clinical trials, then where has all that effort really gone? So having things like a contact registry to be able to connect patients to clinical trials they may be eligible for is super important.
And so I'm going to turn to you, Ashley. What advice or tips and tricks do you have that keeps the community engaged to be able to participate and continue to participate?
I mean, I think that's kind of what we all are here for as patient advocacy groups, right? To keep our communities engaged, to be there and be supportive for our communities. And so I think that's why we hold, monthly connects where just families get together on Zoom and just talk. And sometimes there's, a topic, new families or families with epilepsy. And other times it's just, hey, let's talk and hang out. Holding patient advocacy summits. Yes, it's great to get together to do the research and be together and learn, but it's also good to just sit around a table and talk, or have a dance party with your kids. And so those are some fun ways to keep everybody engaged. We also, ask our researchers to do webinars where they can get on and, explain their research. And sometimes we have 5 people on, and sometimes we have, 100 people on. But we always record them and leave them on YouTube. And then families can, when they get that new diagnosis, they can be that family that scours the website or scours YouTube and finds every piece of information they want. They can see these videos and see kind of what's going on. So it's just, I think we also took, about 2 years ago, we made a very concerted effort. We hired a communications person to come in and really focus on getting our website perfect, getting it really up to date. And then also on our Facebook and our Instagram, on our emails, really putting out there as much information as we can and being very transparent about it. We don't have $5 million. We are run solely by volunteers. So just really letting everyone be part of the community who wants to be a part of it in their own way.
And can I just add that the patient foundation involvement is really important. When most companies, I think, look for new targets, that's actually one of the criteria they look for, an engaged patient community, because that helps them find out what's important to them. It helps them find out how many patients there really are. I mean, it's a little bit selfish, if I'm being completely honest, but it's one more thing that you can do to be helpful.
So this looks at all of the different subcategories that we've reviewed thus far, and gets to the total score for KDVS, which we calculated as 22. And anecdotally, most of the genes that we've looked at so far have fallen within this range. Some are a bit lower, some are a bit higher. While you can calculate a number here, we don't want to place too much emphasis on what that exact number is. In isolation, it's a bit arbitrary. The real benefit we think of this framework is just a place to start to begin thinking translationally about your disorder, given that it hopefully includes as many as the steps that it takes to become clinical trial ready. Obviously, it's not a perfect framework, and no framework ever will be like exactly what you need it to be, which is why we're looking into making revisions and updating it. But yeah, the emphasis isn't so much on the score. While you do have the score, though, this could potentially be used, at least hopefully from a PAG perspective, if this is a tool you want to use over time to potentially track your research progress and ideally see that number increase over time, we do think that that is one way the number generated from the score could be useful.
And I think it is from a PAG perspective. Like, I think it highlighted in there that we don't have our endpoints and our outcomes. And that's where an area we're lacking. And we've all heard yesterday's talk with Terry Jo that, endpoints and outcomes are what we need. And Matt, I'm sure, agrees that we need to figure those out. And so that's what we're missing. And looking at that, it's another tool. We pair it with our research plan, gene target. Okay, these are all tools that we can use to guide us and hopefully get to that treatment.
That's actually exactly what I was going to say. It's like you said, Ray, not focusing on the actual total score number, but really using it as a starting point to see where are the gaps in your research strategy that you could use this score to really figure out where to spend those research dollars to really boost a particular category that's in gene target. So, yeah.
And we've been using it more as a tool for an academic research institution to help us prioritize which projects we want to work on, and then also specifically with projects that we like to partner with patient advocacy groups on. It's a way for us to see how the resources we may have at Boston Children's could best support what the PAG's needs are in terms of what their research pipeline looks like at that time. So we hope that there's multiple applications to this, both within the PAG space and then also on the academic research side as well. So just some future directions on where we're taking gene target now. As I mentioned, we're currently updating and revising kind of the scoring schema to generate a gene target 2.0. We are actively working on putting that paper together, and hopefully it'll be published sometime in the near future. As I mentioned, we're exploring what potential uses this framework could have both in academic research centers as well as for PAGs as well.
So having gone through this gene target framework, I'm going to pose a question to both you, Ashley and Matt. What do you think, having gone through this for KVDS, say, how helpful was it to really hold down your research strategy? Do you have any ideas of any things that are still missing that perhaps you would like to see in perhaps the next version? What are your final thoughts on gene target and how helpful it was?
Yeah, as I said, I think that it definitely showed the holes that we have, like endpoints and outcomes. That's a big one for us, and we really do need to get there, and we are working towards that now. But I do appreciate kind of how Ray says, like, it's not just the score, it's everything that's contained in there, and it's nice to have an outside party review it, because I think we're perfect and we're doing nothing wrong, right? But it's nice to have somebody like an expert researcher, genetic counselor, and all that whole team come in and be like, well, actually, you kind of need this, you need here, and you need here, but you're doing really well there, and you're doing really well there. It really, that third party reviewer really does help.
So I would add, I think it's what you guys have all said. It's really good to give the PAGs a playbook, so you can go to a VC or a company and you have all the stuff together that they're really going to care about. So I think that's incredibly useful. Feedback wise, I would say two things. First is, I know you guys have all talked about the score, but the score for your disease might just be low based on some of those, and I wouldn't let that get you down, because it's what it is. And so you just have to think about how you can take whatever strengths you have to convince somebody that they should be working on this. The other is I would really invite people to revisit this maybe once or twice a year. And an example I'll give you is two of the groups I work with, again, one, the Koolen Syndrome Research Foundation, and the other is the Coalition to Cure CHD2. They have big genes. They don't fit into AAV. And 5 years ago, I would have told them to not waste their time. In the last year or so, the technology really has developed where you can deliver a larger gene and probably be relatively successful. And so if you didn't revisit that, you might take a modality off the table that really is worth exploring.
No, and I appreciate hearing that, because that same thing, I years ago talked to somebody and they said, oh no, you're too big for AAV, that'll never work for you. But now to hear that it's things have changed, right? Science is constantly changing. It's good news.
I think that's actually really the point of this session as well. I wanted to highlight your disease or your disease group might not be the perfect candidate for gene therapy right now. And when I say gene therapy, I do want to make the caveat, we're not focusing on any particular modality, of course. We're just talking about genomic-based therapeutics, however wide that spectrum is. Your disease might not be ready right now, but exactly like Matt said, and all the other sessions that we attended today, the technology and the science is really progressing, and you as a community and as community leaders want to have laid the groundwork now, knowing your disease and understanding the genetics and knowing the natural history and all of the other things that we walked through. You want to lay the groundwork now so that if your disease is not amenable to gene therapy now, in 2 years, in 5 years, in 10 years, whatever, you wouldn't hit the ground running when it is amenable. And something like Gene Target is a really actionable way to figure out, right, where are we in our research strategy, where are the weaknesses, what can we do now to really get ourselves ready.
And I would just add, a lot of what's in here is actually modality agnostic. So you're going to need the same information no matter how you're thinking of treating something. And so it will be good information for you to have anyway. It will also help you evaluate what modalities should you be thinking about. If you have a small window, maybe a 10-year, odyssey into gene therapy isn't a good idea. Maybe you have plenty of time and you have plenty of money and you can do ASO, small molecules, gene therapy and everything. But answering these questions will help you understand that.
It's brilliant. And I've been told by my wonderful facilitator that we are getting close to question time. But Matt, I'm going to put you on the spot for just a second. Having gone through this, from an industry's perspective, there are other things that we haven't covered that perhaps are important for communities to know that would make a particular disease attractive to industry. Are there any others that you can share that we haven't sort of covered now?
I kept thinking about that and I couldn't come up with much. But I would just say two things. One of them is, at least the work that I've done, what's critical is showing clear disease biology. So, for all of these things, it kind of gets back to that. What is the therapeutic window? What is the gene in question? How big is it? All those sorts of things. If you can go to a company or an academic group and say, we know this is what's happening, this is being turned off, this is not being transported, that really helps them, and it saves a ton of time. So that's part of it. I also forgot the next part of this time too, so.
That's all right.
I guess what I would add on to that is I've helped, and I know tons of people in this room have helped different groups put together different roadmaps and that sort of thing. What's great about this is instead of that being bespoke every single time, you guys have put a framework, obviously, to that allows everyone to start it. And maybe that means they can either go to an expert, like Ashley said, and say, this is what we put together. And maybe it also saves them money of not having to have someone do it from scratch. So I think it's really a wonderful addition to the field.
Agreed. I think we might be ready for questions. If anyone has questions.
Oh, so you two have to share. I'll say while she's walking that as a young PAG, it's always hard to know, where do I go next? What do I do? I mean, you come here and you're like, oh my gosh, I don't know where am I supposed to be? There's so many great things to do. So I think this helps guide that. And I would also suggest, our relationship with the Boston Children's and Maya Chopra and her team all came from a meeting like this to say, she was on stage and I talked to her afterwards and said, hey, can we talk a little bit more? And she said, absolutely. And she sat down and we had a great conversation, and that's continued that relationship. So as with everything, you form those relationships, go talk to that person, track them down as they go to the bathroom, and get the information you need.
That was a little bit of my question of
Sorry.
deciding where to go with your particular rare disease. I actually had a two-part question. You, Ashley, mentioned that you had an outside group that you brought in to help identify your gaps in the studies or your knowledge. How did you find those people?
I spoke to her question was like, how do we find that third party to come in and review our research plan and create a research plan for us? And I talked to people who had done it before, and I talked to Mitkus and Gap, and I talked to STXBP1. I talked to the big dogs and said, who do you guys work with? And they said, you need to join Combined Brain. And as a rare neurodevelopmental disorder, we wanted to join. And they were like, we're not taking anybody right now, but you could do the Rent a Neuroscientist program. And so we rented a neuroscientist, and she and her team went through all of our research and looked at everything, and then came up with our research plan and found our holes and gaps.
Okay, that's awesome. We just joined Combined Brain, so
Perfect.
That's a good start. And then how did you get to Boston Children's? Like, how do you find the academia or whoever that's going to pick up your rare disease and run with it?
Excellent question. For me, just keep talking to people. I mean, have you done the Ultragenyx boot camp? That was fun. You guys still doing? Yes, okay.
Twice a year.
Twice a year. That's great. I mean, you talk about overload of information. You go there and you just are like, wow, okay, and you learn from experts and all kinds of amazing people. They even teach you how to give a speech and how to, give your 30-second elevator speech, and it's very important. And talking to those experts, and then coming to these conferences and talking to everybody on the stage, like, we have this, we have that. And same thing with Maya. Like I said, she was up on stage, she got off afterwards, and I said, hey, I had a question about this, and she was like, what organization are you with? And I told her, and she goes, oh yeah, I've seen that a few times. I'm like, what? You've actually seen it in person? What? And she's like, yes, I've diagnosed a few people with that. And from there, I was like, oh my gosh, we have to keep this relationship going. And then also, we hired a chief scientific officer part-time. And she's been helpful in connecting me with other groups of like, oh, they're doing this, you guys should do that. Or you're a chromatinopathy, you need to work with other chromatinopathies. And really making those connections, that's been beneficial. So it's kind of a bit of outsourcing, but I don't know what I don't know. I mean, this is not my field, so that's how I found that it's helped us. I feel like it's a good use of money. It's part-time money, it's not full-time staff, but.
Let me just add two things while you're walking. One is, if anyone is interested in the boot camp, let me know, because I'm part of the organizing group and I can help you, determine if it's a good fit. The other is, I would echo what Ashley is saying, is don't be afraid to ask people. You'd be surprised how many CEOs and other people will respond to you if you just email them. So I'd say give it a go.
I'm just going to add one really quick thing, one very basic thing that you can do, and it actually does yield a lot of results, is finding researchers through publications, peer-reviewed publications. So literally opening up PubMed, finding the recent papers, and contacting the lead author. And as a scientist, I can tell you, most of us will gladly send you a PDF of our publication, because not very many people are asking us to share our publication because it's so scientifically dense. But if you just contact lead authors, they might not be able to answer your question directly, but they could recommend other scientists who could act as scientific advisors for you. So it's not, I really want to stress, don't underestimate contacting researchers, scientists directly through their publications. It happens all the time where you get a lead and you get another lead and it leads to something else.
And don't get discouraged if you contact them and the first time they don't respond. Contact them again, or contact them a different way, or you hear they're speaking at a conference, go talk to them. They may, we miss emails all the time. They may have missed the email.
Hi, this is Gokul from India. My question is to Matt. We belongs to CDKL5 South Asia community. I'm a father of a CDKL5 kid. Could you please give an update about UXO55? And second question, like, as a parent organization from India, so we have close to 450 kids with CDKL5, and every day we are updating the new kits, and we have recently created a Center of Excellence in India. So how as a parents organization we can contribute to that therapy or in terms of support, please?
I'm really sorry, I didn't fully understand the question. Could you repeat it or maybe say it in a different way?
Here. Yeah, maybe I'll try to help a little bit. So he has a child with CDKL5 deficiency disorder and is looking for some updates generally on the program, but then also has established a center of excellence in India. And I think maybe you could speak more broadly to centers of excellence globally outside of the United States, because many organizations here have participating groups in other countries. It might not be India, but other countries, and like how to really best work together with those sites outside of the United States.
Yeah, and I think some of that is exactly what Ashley said. Don't be afraid to reach out to people. So for CDKL5, the Lulu Foundation is huge, but they don't have a presence in every part of the world. I'm sure they would be glad to partner with you, because that having another center of excellence, right, allows people then to do trials there. So I would just really encourage you to reach out to people.
Sir, I have posted one more question, like Ultragenyx is doing a gene therapy program for CDKL5. Could you please update on that?
Oh, yeah.
Yeah.
We can chat.
Maybe-
I'll be honest, I probably can't tell you anything that isn't publicly available, but maybe I can send you to some people that can give you an update.
Yeah. Other questions?
I actually had a quick answer to the first question too. There's been a lot of conversations today about grouping, lumping and splitting. And one way to potentially find some researchers who may also be interested in your condition is to reach out to patient advocacy groups who have disorders that either are similar in their biology or similar in their clinical features, and see if they've been able to connect with other researchers. And at least I can speak to our experience at Boston Children's. Our PI is interested not just on one particular neurodevelopmental disorder, but those who share a similar biological mechanism, and studying those conditions as a whole. So you may also have some luck in just connecting with PAGs who represent similar conditions to yours, to see if they've met with other researchers who are interested in their condition.
Yeah, and sometimes you can get researchers interested in a new disease area because they didn't know it existed, and they'll be like, hey, I look at the Golgi all the time, I didn't know this was a thing, it's a really interesting phenotype. So I think that makes sense to look at the researchers in the area and see who you can network with.
And again, the whole, don't get upset if at first you don't succeed. We applied for a RARES 1 CZI grant with a bunch of other chromatinopathies. Didn't get it. But, we're going to keep trying and keep trying and go back to all different ideas. We always have, 10 different things we're working on at the time. And if we get one of them out of 10, then we're doing a pretty good job.
So I'm going to ask a question since we have a little bit of time. In work that I do, and we at Global Genes are doing with lots of patient advocacy organizations. One of the gene target items was reversibility in a model system, right? And one of the questions we frequently get is it okay just to have cellular models? In a lot of disorders, it's very, very difficult to get a mouse model to either recapitulate the phenotype, so to look like what it looks like in human kids, or sometimes to even have the mouse survive. So thoughts on that question? It's a very common question that I hear.
Yeah, so I think it's really common, right, for any animal model not to recapitulate everything. And sometimes it seems that to be the thing you care about, right, is the thing that it doesn't recapitulate. So I guess two things. One of them is if you can use a cell system and get at the basic biology, that can be really useful. Again, if you know something, you fix trafficking or you fix an interaction or whatever that is, I think that goes a long way to saying we know what basically causes this, and it looks like we're doing something to that very basic thing. I think the other is if you don't have an animal model, but you really understand what's going on in the patient, that can also be really useful, because maybe it'll help you extrapolate what's going on in your cell model, or even just make very compelling arguments as to why it's reversible. So there are definitely ways around it. It's just ideal if you can have everything. The other thing you could do, and this is only if money is, infinite, is you can continue to make models, and maybe not all mouse, maybe they're a rat or whatever, a rabbit, to keep trying to get that phenotype. Sometimes you're chasing your tail, sometimes it works. So I wouldn't, say definitely do that, but, engage your SAB and see if there are things that others have done.
And to that point about making the different models, yeah, we've got mice and zebrafish and frogs and all that, and it's all very interesting. But as Ray said, we have 60% of the microdeletion and 40% have the variant. So therefore, we have to make 2 of everything because we need to have it in both ways. As much as we say it's the same, is it really the same? And we won't know that unless we test.
And I do also want to say it could be that different animal models are showing a totally different phenotype, and how you correlate. So animal models, will be great, but again, with the caveat that, a mouse model might show a phenotype that might be completely different and might be confusing as to what maybe a zebrafish is showing. So having to parse that out is important.
Any other questions? I think we have time for one more. Yep, one. Is there a place where we can find these resources?
So these should all of these slides should be in the event app, and these should all be, you should be able to click on these and find these. If you have any problem finding them, please just email Global Genes. I'll be happy to send this to you as well. Yeah.
And the paper is publicly available too.
And the paper is the second link here. It's publicly available.
Okay, well, thank you. Why don't we all give a round of applause?